Hello and welcome. I am Francesco Reina and this is my poster submission for the session Advanced Optical Microscopy at the EBSA meeting 2021. In this work, we employed interferometric scattering microscopy, or ISCAT for short, as our technique to investigate lipid diffusion on the plasma membrane. In ISCAT microscopy, the contrast is generated by interference between reflected and scattered field, which means that the signal we detect is directly proportional to the incident light intensity, making high-speed tracking possible. However, large scattering tags are necessary in the majority of cases. In order to probe lipid diffusion on the plasma membrane of PTK2 cells, we fed the cells with a biotin-related lipid analog, the SPE-PEG biotin, which we then labeled with streptavidin-coated gold nanoparticles of two different sizes. Thanks to ISCAT microscopy, we were then able to track the diffusion of these lipids with a frame rate of 2 kHz. Instead of using the mean-squared displacement to quantify the diffusion motion of the lipids on the cellular membrane, we used the motion blur-corrected apparent diffusion coefficient, the AP that we see in the slide. We then fitted three models of diffusion to our apparent diffusion coefficient data. One is free diffusion, in which the particles move in an unrestricted way on the surface. The second one is the confined diffusion, in which the particles can only diffuse within a certain area of the cellular membrane. And the third one is the compartmentalized diffusion, where the particles are only transiently confined and then jump from one compartment to the other on the cellular membrane. The most appropriate model to describe the diffusion is then selected through minimization of the Bayesian information criterion. In this slide, we see the results of our analysis protocol applied to the ensemble average apparent diffusion coefficient data obtained from the trajectories of gold-tagged lipids on the PTK2 cellular membrane divided by gold nanoparticle size. We notice how the long-range diffusion coefficient, the M, is much lower for the 40 nanometer gold nanoparticle than it is for the smaller 20 nanometer gold nanoparticles. However, the other parameters, which describe the transient confinements felt by the lipids on the cellular membranes, are largely invariant with respect to nanoparticle size. In order to validate our findings, we compared our experimental data with relevant simulations. We simulated the diffusion of a point-like particle on a corralled environment where the particle has a probability p-hop of jumping across compartments, thereby generating something similar to the compartmentalized diffusion we observed. We now define a parameter called confinement strength, which we use to quantify the effect of the compartmentalization of the cellular membrane on the diffusion of molecules on its surface. This parameter is defined as the ratio between the diffusion coefficient at time 0 and the diffusion coefficient for time that goes to infinite. In the case of the compartmentalized diffusion model, this is effectively equivalent of the ratio between the sum of the mu and the m and the m by itself. In this graph, we plot the confinement strength against the probability of hopping of the simulation executed as we explained before, the new data, that is the data from this study, and data from other studies from this group. For the experimental data, the p-hop is estimated as the p-hop of the corresponding simulation that most closely resembled the behavior of the data. From our experimental results and their accord with the corresponding simulation, we can say that we confirmed the compartmentalized nature of the cellular membrane, we obtained a framework in which we can compare diffusivity results across techniques and different experiments, and we can quantify the physical characteristics of the cellular membrane according to this model. Another significant advancement in single particle tracking on the cellular membrane would be to forego gold nanoparticles entirely and instead track single fluorescently labeled lipids with kilohertz sampling rates. Unfortunately, we don't have enough time to go into the details of single particle tracking through MINFLUX. Suffice to say that we've been able to perform single particle tracking on single fluorescently labeled lipids embedded in a supported lipid bilayer with sampling rates around 8 to 10 kHz and localization uncertainties in the 10 to 20 nanometer range. Please notice that the localization uncertainty that we report here is related to the analysis that we performed before and not to the theoretical resolution limit of MINFLUX. Thank you for your attention. I would like to acknowledge the support from the Biophysical Imaging Group in Jena led by Professor Eggeling, give special thanks to my collaborators and of course to the funding bodies, and point your attention to the video description in which you will find the links to related literature and materials. Thank you once again.